Hello students, welcome to EPG Pathshala. I, Dr. Vikas Rana from Punjabi University Patiala. We are going to talk on module Pharmaceutical Applications of Microemulsion from your paper Novel Drug Delivery System Part 2. During the last 2 decades, microemulsion have been the drug delivery system of choice due to some reasons, that is thermodynamic stability, optical clarity, particle size is less than 200 nanometer, ease of penetration is very high, solubilize large number of lipophilic drugs. You know, there are large number of lipophilic drugs these days are coming into the market, but they are having low bioavailability. So it has an ability to increase the bioavailability of these lipophilic drugs. And more, one of the very important part is patient's compliance and acceptability of these microemulsion delivery systems. So students, let's see what are going to learn from this module. The main objective of this module is to know different pharmaceutical applications of microemulsion based delivery systems. In one of the another objective is we are highlighting various research activities in current in the development of microemulsion based delivery systems. Oral microemulsions are beneficial in enhancing the solubilization of poorly water-soluble drugs, particularly the drugs which belongs to category of BCS class 2 or BCS class 4. They overcome the dissolution-related bioavailability problems. Another advantage is the oral microemulsion contains polar, non-polar and interfacial domains. The hydrophilic drugs and macromolecules can be encapsulated with varying solubility. In addition to it, these oral microemulsion systems protect the drug from oxidation, enzymatic degradation and enhance membrane permeability. The main purpose of microemulsion based drug delivery system is to increase the solubility of a drug. These are the different examples which will show that the drug, even if given by parenteral route or topical route or transdermal route or dermatological route, it is possible to increase the solubility of a drug employing microemulsion based drug delivery system. In another example, the microemulsion is also associated with increase in permeability or enhancement in the permeability of most of the drugs like apomorphine hydrochloride, ketoprofen, diclofenac, and as well as that of the estradiol. They are also known for enhancing the bioavailability of microemulsion such as dexamethasone, sumatriptan, and the stability of most of the drugs are increased when incorporated into microemulsion based drug delivery system. For example, doxorubicin, progesterone when given in dermal route. In another examples, the microemulsion based drug delivery systems are also suitable for better absorption, enhanced drug dissolution and drug bioavailability. For example, it help us in increasing the bioavailability of glipizide when given in oral route. Therefore, these delivery systems are effective in oral based microemulsion systems, and it improves oral bioavailability of hydroxycephalur yellow A, and they are also known for increasing solubilization, patient compliance of Acyclovir topical delivery systems. Therefore, on this basis, we could conclude that there are a lot of applications of microemulsion based delivery system in different areas of the pharmaceutical preparations. We can divide For our study purpose, these preparations which are prepared in the literature into different categories, like microemulsions which are prepared for oral delivery, microemulsions prepared for topical delivery, microemulsion prepared for ocular delivery, pulmonary delivery system, nasal delivery system, periodontal delivery system. Apart from that, microemulsion based delivery system are also effective for drug targeting, like brain delivery, that is drug targeting to brain, drug targeting to lymphatic system, drug targeting to tumor cells, as well as drug targeting to cellular targeting system. They are also useful in these areas. So keeping in view, now let us discuss what is the role of microemulsion and what are the formulation aspect to be changed for the delivery of such drugs through these routes on basis of microemulsion based delivery systems. Therefore, as a first example, let us discuss microemulsion for oral delivery of drugs. Therefore, conventional ophthalmic dosage form that contains water-soluble drugs are delivered in aqueous solutions as eye drops and eye solutions, whereas water-insoluble drugs are formulated as suspensions or ointments. Therefore, these microemulsion based ophthalmic preparations, when prepared by this particular technique, they can solubilize the water insoluble type of drugs, that is lipophilic drugs, and therefore are quite useful for this particular purpose. Thus, the main problem with conventional ophthalmic delivery systems have low corneal bioavailability, lack of efficiency in the posterior segment of ocular tissues. Therefore, the examples of microemulsion for oral delivery system which are commercially available are Neoral R, which is of cyclosporine A, Photovasa R, which is Sequenavir, and Norvir, which is Ritonavir. Therefore, this image shows comparison of cyclosporine pharmacokinetic when given in emulsion as well as in microemulsion dosage forms. You can clearly see that when the cyclosporine is delivered in microemulsion based system, the Cmax is just 200 units, that is nanogram per ml, whereas when new oral, that is microemulsion based system of cyclosporine is given, the Cmax level rises to 1000. These indicates that these microemulsion based delivery system enhanced the bioavailability of drugs. Now let us discuss dissolution profile of microemulsion based delivery system versus emulsion formulations. The dissolution profile, if we have seen that the percentage release of Acyclovir from microemulsion based delivery system is around 90% in just 4 hours, whereas plain drug solution is just 30% is released in 8 hours. In another phase of this study, the percentage diffusion versus time plot indicate that around 90% of drug diffuses in just 300 minutes. Thus suggested that dissolution profile of microemulsion based delivery system, for example Acyclovir, is enhanced. Both these findings indicate that microemulsion enhanced solubility of Acyclovir. On comparing emulsion or tablet based delivery systems of Acyclovir with that of the microemulsion, the results suggested that plasma concentration of microemulsion based delivery system is quite high, around 1.6 microgram per ml when given in microemulsion based delivery system, whereas in tablet formulation it was just 0.8 microgram per ml. In a similar way, when emulsion delivery system compared with microemulsion delivery system, the Cmax level increased to 15 microgram per ml as compared to those in emulsion delivery system, whereas they have just 8 microgram per ml. Thus, the results suggested that microemulsion prepared with SPAN 83 and SPAN 80 are quite effective in enhancing the bioavailability of Acyclovir. In another example, dissolution profile of tablet is compared with that of microemulsion formulations of Valsartan. The in vitro release profile of Valsartan from different core in cup tablet formulations in 0.1 molar HCl, when examined, the results suggested enhancement in dissolution performance of Valsartan. Whereas when these delivery systems were given in the form of microemulsion, there is a remarkable increase in dissolution performance as compared to that of the tablet. Thus, the finding pointed that there is increase in dissolution profile of Valsartan when given in microemulsion delivery system when compared with that of the pure drug to that of the tablet delivery system. The pharmacokinetic performance of oral suspension of Valsartan, the Valsartan when entrapped into gel formulation, and Valsartan when given in solid self-emulsifying drug delivery system were examined as compared to that of the pure powder. The results suggested that microemulsion based delivery system are quite effective in enhancing the bioavailability of Valsartan. If you see the correlation between plasma concentration of Valsartan with the time of different delivery system, the Cmax levels of oral suspension was 1400 nanogram per ml, whereas from that of the gel delivery system it was just around 7000 nanogram per ml. And leaving these comparisons, you can see there is a remarkable increase in Cmax level to 4 microgram per ml of Valsartan when loaded into SMEDDS delivery system as compared to less than 1 microgram per ml in case of pure drug. Thus, the findings pointed towards the bioavailability of Valsartan microemulsion gel and SMEDDS formulations when compared with oral suspension or with that of the pure drug. Now let us discuss microemulsion for ocular delivery. On comparing the microemulsion based delivery system with that of the conventional eye drops, it is evident that the microemulsion based delivery system are thermodynamically stable. They are simple and inexpensive, and they are industrially acceptable for the production purpose, as well as they are sterilizable. That is the reason that these microemulsion based ocular delivery system are preferred nowadays for water insoluble drugs also, which are earlier given in suspended form. Now, microemulsion when prepared for parenteral delivery system, they are a very difficult task to provide lipophilic and hydrophilic drugs in microemulsion based parenteral drug delivery. Oil water microemulsions are most effective in the parenteral delivery of springly soluble drugs. Frequently administration of drugs can be avoidable by administering high concentration of drugs entrapped in the microemulsion. So, in comparison to lipophil liposomes or other vehicles, it exhibits a high physical stability in plasma. Internal oil phase is more resistance against drug leaching. Thus, for the developing parenteral microemulsion, the main ingredients which generally influence the permeation across membranes is C3, C4 alcohols. They are replaced with parenterally acceptable co-surfactants like polyethylene 400, polyethylene glycol 660, 1,2-hydroxystearate or ethanol type of co-surfactants are quite useful. The main example of microemulsion formulations of drugs prepared to enhance solubility are fluorbibuprofene and glimepiride. Example of microemulsion formulations of drugs prepared to enhance bioavailability is itraconazole. Thus, to improve effectiveness and avoid drug delivery related barriers, microemulsion based ophthalmic delivery systems are prepared. For example, chloramphenicol eye drop is prepared for trachoma and keratitis. These delivery systems, chloramphenicol based delivery system consisting of span 80, span 20, tween 20, isopropyl myristate and water based eye drops when prepared show remarkable increase in stability as well as that of the bioavailability, thus suggesting that the chloramphenicol eye drop on microemulsion based chloramphenicol eye drop is quite useful for the treatment of trachoma and keratitis. In another example of dexamethasone eye drop, it was evident from these study that dexamethasone eye drop prepared by microemulsion based technique is better tolerable, highly bioavailable and greater penetration in the eye. Thus, this suggested that there is a decrease in dose frequency when dexamethasone eye drop is administered and it improves patient's compliance. Therefore, these findings pointed towards utilization of microemulsion based dexamethasone eye drop for the purpose of treatment for various treatments of microemulsion. Now let us discuss microemulsion for parenteral delivery of drugs. The main advantage of administering microemulsion for topical administration of drugs are the effect of hepatic first pass metabolism can be avoided. The second important thing is drug metabolism in salivary. The third is inhibition of drug degradation in the stomach and related toxicity effects. Therefore, microemulsion based topical delivery is the direct delivery and targetability of the drug to affected area of the skin or eyes. Microemulsion for transdermal or dermal delivery of drugs. Periodontal disease is associated with progressive oral pathological afflictions like inflammation, degeneration of the gums, periodontal ligaments, cementum and its supporting bone. It is a major cause of tooth loss. The main problem associated with the existing periodontal products is, that is jellies, ointments or sprays, they lack of efficiency due to inadequate depth of penetration, too short duration, difficulties in administration due to spread, taste, etc. So these are the main problem associated with the existing periodontal products. Therefore, the development of microemulsion based pain relieving gel consisting of local anesthetic in oil form containing surfactant and water was prepared by Brodinil, and we have found that there is a very good response coming out of it. Into this formulation, which is microemulsion based, optionally a taste masking agent was also added, and they had thermoresponsive gelling properties, that is, they are having less viscous at room temperature than after introduction onto a mucosal membrane of a patient. The main surfactant imparted the thermoresponsive gelling property. They are Ploxamer 188, Ploxamer 407. These are the agents which are generally, you can say, added as a thermoresponsive, and these are added into these formulation to get a very good results. But ultimately, the result which we have obtained is because of the entrapment of microemulsion based delivery system. The examples of various attempts to be taken for enhancement of permeation of hydrophilic drugs across skin are: we can increase the permeation of 5-fluorouracil, apomorphine hydrochloride, diphenhydramine hydrochloride, tetracaine hydrochloride, methotrexate. The examples for lipophilic drugs are estradiol, phenasterine, ketoprofen, meloxicam, felodipine, triptyline are the examples of drugs which are show enhanced permeation when given in microemulsion based delivery system. However, the major issue which is related with microemulsion based delivery system for transdermal or topical delivery system are the high surfactant content, because the presence of high surfactant concentration leads to skin irritation. Therefore, when studying such type of delivery system, it is very important to check skin irritation of microemulsion based delivery system. Otherwise, you will not achieve a good property of microemulsion for delivery to transdermal or topical delivery of drugs. The percutaneous permeation of tyrosine through Franz diffusion cell is examined when entrapped in emulsion or microemulsion based delivery system. The correlation of percentage absorbed tyrosine versus time indicate that microemulsion absorbed in higher concentration as compared to emulsion based delivery system. The microemulsion increases the absorption coefficient of lidocaine when compared with conventional emulsion in vivo. The correlation between lidocaine versus time clearly reflects that there is an increase in bioavailability of lidocaine when administered in microemulsion based delivery system. The absorption coefficients also shows more than 700 units increase in absorption coefficient when entrapped into microemulsion based delivery system. Thus, the study suggested that microemulsion is a better than that of the conventional emulsion based delivery system. This image is an image of a marketed product of Eller-Mill, which was jointly developed by Verbeck and Gattifoss, and is first veterinary cleansing product based on the microemulsion. It consists of surfactant labrasol and co-surfactant labroglycol. Therefore, the benefit of microemulsion based topical and transdermal delivery system are clearly evident. In another example, ex vivo permeation of Amphotracin B microemulsion across abdominal skin of albino wistar rat is examined. And interesting findings are visible that all the microemulsion based delivery systems of Amphotracin B remarkably and significantly different from that of the plain drug solution, thus indicated that microemulsion act as a permeation enhancer for Amphotracin B. The correlation of cumulative amount of drug deposited versus time for detected during ex vivo permeation of Amphotracin B in nanoemulsion across abdominal albino rat indicated that drug solution shows lowest permeation and nanoemulsion based delivery system showed enhanced percutaneous permeation of amphotracin B. The marketed formulation Fungisome containing amphotracin B is showing intermediate permeation across abdominal albino rats. Thus suggested, nanoemulsion based delivery systems are quite useful for the delivery of amphotericin B in nanoemulsion formulation. The drug retention of amphotericin B in microemulsion nanoemulsion in abdominal albino rat skin, when examined, this suggested that there is a enhancement in drug retention when given in microemulsion or nanoemulsion based delivery systems. And on comparison with drug solution or fungisome suggested that nanoemulsion has higher and drug retention in the abdominal albino rat skin. This could be quite useful when it has to be given in topical or transdermal based delivery systems. And another example of Grisiofulvin, when ex vivo permeation of Grisiofulvin microemulsion across dorsal skin of laca mice was studied, the comparison between microemulsion and ethosome was conducted. The results suggested that microemulsion based formulations shows highest permeation across dorsal skin of laca mice and compare with that of aqueous suspension, liposomes, ethosome gel, ethosome dispersions. The drug retention behavior of Grisiofulvin microemulsion in dorsal skin of laca mice also revealed that microemulsion based delivery systems are quite successful because of their high drug retention capacity. Let us discuss microemulsion for periodontal delivery of drugs. Now, the more LDL receptors in the tumor cells lead to destruction of the tumor cells. Thus, higher concentration of anti-cancer drug could be achieved in the neoplastic cells that have an increased expression of the receptors. In this way, toxic effect of these drugs on the normal tissue and organs can be avoided. Microemulsion for targeted delivery of drugs. Neoplasm cells have an increased number of low-density lipoproteins receptors compared to normal cells. Therefore, LDL is equal to lipid plus protein portion. And the microemulsion which is used for this particular purpose, that is drug targeting, consists of nucleus of cholesterol esters, not more than 20% triglyceride surrounded by a core of phospholipids and free cholesterol. A chemotherapeutic agent is added. Therefore, the above microemulsion is equal to only lipid portion of LDL and not that of the protein portion. LDL receptor consists of lipid portion plus protein portion, and lipid portion is equal to that of the microemulsion. And into these microemulsion, to cover up the deficiency of protein portion, lipoprotein E, that is ApoE, is added into the microemulsion. This ApoE acts as a linking element with that of the LDL receptor. And this LDL receptor as well as the microemulsion is acting as a linking element between the particles of the microemulsion and the LDL receptor. This whole of the unit is allow microemulsion to enter into the cells via LDL receptors. The LDL receptors are quite higher in case of tumor cells as compared to the already conventional cells which are present, that is normal cells which are present in the human being. Therefore, the increased number of LDL receptor leads to increase in permeation of more amount of microemulsion containing chemotherapeutic agent. This results in effective delivery of drugs to the cells. Therefore, the main purpose of microemulsion based delivery system as observed from the literature, that for example fluorbibuphine was given parenterally and we have found that there is increase in solubility. Another example is ibuprofen given in parenteral form was found to have increased solubility. Ibuprofen also given in topical formulation showed its increased solubility behavior. Apart from that, if aclofenac is given dermatologically, there is increased solubility. Chloramphenicol given in ocular delivery system was shown to have increased solubility. Therefore, there is a lot of advantage of microemulsion based delivery systems. Apart from that, when we talk of transdermal based delivery systems, drugs like apomorphine hydrochloride, ketoprofen, diclofenac, terbutaline, languistragine, phosphate, estradiol, these drugs are found to show enhanced permeability or enhancement in permeability characteristics. That is why these drugs are now preferred for you can say microemulsion based delivery systems. Apart from that, another example for the ocular delivery is dexamethasone or sumatriptan, which are having enhanced bioavailability characteristics. Uh, in addition to it, we have a drug like itraconazole, which are given parenterally for a better absorption. Timolol, which is given in ophthalmic preparations, which has a better absorption. So all these properties of the drugs make suitable for enhanced bioavailability characteristics. In an example, Shiokawa and co-workers developed folate-linked microemulsion of aclasinomycin A. These folate-linked microemulsion was found to be feasible delivery system for tumor targeting, and the study showed that folate modification with sufficiently long PEG chain on microemulsion is an effective way of targeting emulsion to the tumor cells. For the purpose of brain targeting, intranasal administration of microemulsion based delivery systems are to be given. These intranasal administration is quite useful in terms of its simple, practical, cost-effective, convenient, and non-invasive route of administration for rapid drug delivery. A direct transport of drugs to the brain circumventing the brain barriers is the main advantage of delivering microemulsion based delivery system through the nasal route. This image indicate that how microemulsion is helpful in transferring the drugs to that of the brain. From the nasal delivery into the olfactory lobes, the microemulsion are transported to olfactory bulb. From the olfactory bulb, these microemulsion containing drugs are transferred to olfactory tract, and from these olfactory tract they are directly reaches to the brain. That is the reason that the microemulsion based delivery system, when given through intranasal route, are quite useful. The paracellular transport, transcellular transport, and active p-efflux inhibitors activity of microemulsion leads to improved brain targeting, and it can easily cross blood-brain barrier. This is the reason that intranasal delivery for the brain targeting in the form of microemulsion delivery system is quite useful. As an example, the drugs which are targeted through intranasal administration into the brain. The examples of such drugs are clozapine, clamamabgine, sumatriptan, diazepam, lorazepam, alprazolam having log p-value near to 1.5 to 3.5. Risperidone, buspirone are the drugs which has extensive first-pass metabolism. Norbiletin has systemic toxicity. Such type of drugs are generally delivered through this route by formulating them into microemulsion based delivery systems. When nimodipine was administered through different microemulsion based formulations, and its in vitro performance as well as ex vivo performance was examined, the results indicated that the order through which the nimodipine released from the respective formulations are: the lowest was from suspension, followed by a gel, that is mucoadhesive microemulsion gel, and then with that of the nimodipine microemulsion. On comparing its ex vivo performance, the order is first nimodipine suspension, then increase to nimodipine microemulsion, and then highest through in situ gel, which is mucoadhesive microemulsion. Thus suggested that microemulsion based delivery of nimodipine is quite successful in enhancing the bioavailability of nimodipine. When nimodipine was administered through intranasal route in the form of microemulsion, nimodipine suspension, or in situ gelling in mucoadhesive microemulsion, it was evident that maximum collection of nimodipine was at the brain and lowest was in the plasma. These findings pointed that nimodipine, when given in emulsion formulation, has maximum concentration, although all of the amount of total drug, irrespective of the type of formulation, it was higher in case of drug administered through intranasal route to the brain. However, the brain delivery, brain targeting is only possible when given in microemulsion based delivery system. Therefore, approximately around 2800 nanogram per ml drug was retained in brain as compared to lowest amount in plasma, that is around 250 nanogram per ml. Thus, these findings pointed towards successful delivery of nimodipine when administered through intranasal administration as well as in the form of microemulsion based delivery system. In another example, where the permeation of Udenafil microemulsion across H&E cell monolayers were examined, the results suggested that microemulsion based delivery system shows maximum permeation across these cell lines as compared to the control samples. Among these microemulsion based formulations, the main composition consisting of water 45%, Capmul MCMCl8 15%, Labrasol 20%, and Transcutol 20% is considered as the best formulation of Udenafil microemulsion. A similar microemulsion based Udenafil formulation, when given to rat models, the plasma concentration of Udenafil versus time was plotted. The results suggested that IV route follows higher Cmax as compared to oral administration, and Udenafil solution after intranasal administration is less than that of the microemulsion based formulation. Therefore, these suggested that intranasal delivery is successful for the Udenafil microemulsion delivery to the rats. So students, let's summarize what we have learned from this module. Microemulsions are the powerful technique for the formulation of poorly water soluble drugs for different routes of administration like oral delivery, dermal, transdermal, topical delivery, periodontal delivery, ocular delivery, targeted delivery, especially intranasal delivery, which are directly delivered drugs to that of the brain. They are called as one of the very good advantageous tool, powerful tool for the administration and formulator. As a formulator we have. Thank you.